Hello, I'm Sandra Moreno from University of Bristol, and I'm presenting testing the ovarian ground plan hypothesis for the evolution of sociality using the solitary wasp Ammophila pubescens. One of the major transitions in evolution is the transition from solitary to group living, and this transition has been extensively studied in Hymenoptera species, and among the most prominent hypothesis of this evolution is the ovarian ground plan hypothesis. And it states that different phases of the ovarian development of the solitary insect's nesting cycle regulates the reproductive and non-reproductive phases of its cycle. These alternating states could be a mechanistic readaptation for the evolution of the worker-like and queen-like caste in social insects. And this has been tested in primitively and highly social species in Hymenoptera, and to the best of our knowledge, tested once in a solitary Hymenoptera. So here, we use the simultaneous progressive provisioning wasp, Ammophila pubescens, to test this hypothesis by using a combination of behavioral experiments, gene expression, and physiological measurements, and evaluate whether each phase of her nesting cycle shows similar gene expression profiles as expected in queen-like and worker-like behaviors in social insects, as predicted by this hypothesis. So we studied Ammophila in southwest England in the Dorset area, and there we characterized their behavior and sampled the wasps. And in the lab, we measured the longest oocyte for each behavioral phase and extracted the RNA from the head cavity. The extracted DNA then was sent to China for RNA-seq, and a total of 34 libraries were sequenced. And once we got the sequences, the bioinformatics pipeline went from quality check to differential gene expression and gene ontology. Our physiological measurements did not show evidence of ovarian regulations, even when grouping the phases as queen-like and worker-like. And the evidence for an ovarian ground plan at the transcriptome level was weak, with no functional enrichment. And there were genes differentially expressed, previously associated with caste differentiation in social insects. When we contrast each phase against the rest, we found lack of a clear differentiation of behavioral phases, except provisioning behavior, which was the most distinct phase. And among the genes differentially expressed in this phase were genes involved in sensory perception and immune response. The enrichment analysis showed gene ontology terms overrepresented for the provisioning behavior, and these terms are involved in energy expenditure, which makes sense as these wasps during the day are hunting, stinging, and carrying prey to their burrows or nests. A further comparison between provisioning phase and the rest of the phases show that provisioning behavior is still being the distinct phase, except when comparing with egg laying. However, both differential gene expression analysis suggest that provisioning behavior is regulating the solitary nesting cycle. In conclusion, Ammophila pubescens can be used to test the basis of a mechanistic ground plan for the evolution of sociality in insects. Our results suggest that cycles of provisioning behavior, not ovarian physiology, are the main drivers in gene expression during the nesting cycle of this wasp. And importantly, our results suggest that social castes evolved by co-opting the regulatory mechanisms underpinning the provisioning cycles of solitary wasps, as well as the ovarian cycles. And these processes are likely to be uncoupled in the emergence of queen and worker castes in social insects. Finally, we would like to thank our collaborators who sequenced and annotated the Ammophila genome, to National Trust for allowing us to work in their land, the people who was involved in the field, and the organizers of this conference for allowing me to present my work.